Hi everyone, I will present the subtitle static Raine and a research fellow from QUT Center for Robotics. She recently finished thesis, which is a collaboration between QUT Center for Robotics and Data 61 SIRIS. Well,
Thanks so much for having me here at the University of Sydney. I'm so happy to be here. I came pre-prepared for the dress code today. Today, I'm going to be telling you a little bit about my research. A lot of this work was done during my PhD, but I'm also going to talk about my current work as well, and maybe briefly a little bit about myself at the beginning as well. I'd like to start by acknowledging the traditional owners, of the lands where QUT stands, that's the Turrbal and Yagara people, the people of where University of Sydney stands, the Gadigal people of the Eora Nations, and I'd also like to acknowledge the Quandamooka people, where a lot of the data collected in this work was collected. Okay, so to give you an idea of what I'm going to talk about today and where we're going, just briefly a little bit about myself, then I'll touch on the motivation for my research, and then I'm going to talk about three main bodies of work. So starting off with the problem of multi-species coarse seagrass segmentation. So that's monitoring for different species of seagrass. Then I'm going to talk about coral segmentation. So a similar problem, but here looking for different types of corals in images. And then finally, getting more into that intervention space. So here we're looking at the coral reef restoration problem, and that's work I'm currently doing with the Reef Restoration and Adaptation program. And then at the end, there should be a little bit of time to share some ideas about future work where I think we can go from here. So just briefly about myself, I'm currently a research fellow at QUT in the Center for Robotics. I'm interested in artificial intelligence, computer vision, and robotics, and I would say that my research really lies more at the applied side of things. I'm interested in coming up with new innovative algorithms, but a lot of the time it's because there's a really challenging specific problem in a challenging domain that we need to address. I'm really passionate about environmental monitoring and conservation, so that's why a lot of the work I'll be talking about today is really in that underwater monitoring, reef restoration domain. Okay, so now onto the motivation. So today I'm going to be talking about two ecosystems in particular. First of all, seagrass meadows. These are kind of the less sexy cousin of coral reefs, I suppose, but they're really, really important ecosystems with a whole range of different roles and services. So they support fisheries, they act as a nursery for many different marine species, and they also support marine mammals like the dugong, which is a really culturally significant mammal as well. Now, seagrass meadows are also interesting because they only comprise a really small amount of our ocean, so only 0.2% combined with other types of marine vegetative habitats as well. But in fact, they account for up to 50% of the carbon sequestered by our ocean. So they really punch above their weight in terms of their ability to store and sequester carbon from the atmosphere. The next ecosystem, of course, are our coral reefs, which are a really rich biodiverse ecosystem. The Great Barrier Reef alone accounts for 25% of our known marine species, so really important ecosystems for that reason. They also support our economies through tourism and fishing as well, so they're really significant for us in Australia economically as well as environmentally, but they are under really significant threat due to climate change. There are some projections that estimate even up to 70 to 90% of loss could be seen within the next decade alone. So we do need to place a lot of importance on these ecosystems and really act now to try and save them. Coming from a robotics standpoint, when I learn about these problems and talk to ecologists, I'm always thinking about how can we use technology to improve their work, maybe make it safer for them and what they're doing, make it more efficient, reduce the amount of really manual time-consuming tasks that they're doing. So there's a lot of different problems that I think that we could be using robotics for. A few examples. So on the left there, we've really got the monitoring piece. A lot of traditional marine surveys have been performed by divers, snorkelers, people getting towed behind on boats, trying to see what species there are, trying to remember it, trying to record it. It's, you know, limited in scale, can be quite dangerous work as well, and not super accurate or efficient. So a lot of the work I'll talk about today is tackling that monitoring task. But when we go more into the intervention space, there are a lot of tasks there as well. So on the right side of the frame here, we have some scientists that have collected coral spawn. They're putting them in aquaculture facilities, waiting for that spawn to reach a more mature stage of growth so that we can then use it to reseed the degraded areas of the reef. Historically, they've literally used a microscope to count those coral babies, and they do that every day. And they manually do it with a clipper, and that's just ridiculously labor intensive. So that's another example of a task that I'll talk a little bit about later. So in terms of that marine survey problem, that monitoring task, there are a whole different range of ways that we can monitor or do these surveys. And these different methods have pros and cons in terms of the spatial scale. So that's the area that we can cover with that survey method. And then also the taxonomic resolutions, that the level of detail that we can go down to with that data. So whether we can see different species of corals in an image, for example. I'm interested in playing really in the middle part of the graph. So obviously those robotic methods there, UAVs, ASVs and AUVs are really what I'm interested in, because we do have that good balance, the increasing scale, but still having a high level of detail. So some examples, at QUT, we're doing some drone surveys for turtle monitoring. As the ocean temperatures increase, we have less female turtles being born. So these are turtles coming in to nest, and we're tracking and counting these individuals. The next one here is footage from an autonomous surface vehicle. So that's again for that monitoring task. And then autonomous underwater vehicles as well. So we're also, you know, playing in this space. And then just a few examples of platforms that I've been working with. On the left, you've got your Blue Robotics ROV, very cost effective. And then on the right, this is one of our autonomous surface vehicles. We affectionately call it the floaty boat at QUT. Okay, so just to talk a little bit more about the task and where my research comes in. Obviously, when we do computer vision in typical above water fields, you can usually label images quite easily. So we have image characteristics where we have a clear foreground background separation. We have clear delineation of objects and their boundaries. And it means we have a high annotator agreement. So you can present this image to anyone in the audience, and you'd probably say it's a person on a motorbike, and we wouldn't argue too much about that. And that means that you can then go ahead and use Amazon's Mechanical Turk. You can get a whole lot of images labeled very quickly, pretty cheaply, not a big problem. Then you get into underwater imagery, and I will say, because I know you guys are at Imaging Group, we deal pretty much exclusively with RGB imagery, and that's because it's cost effective, it's available, citizen science groups have it, it's very widespread. So that's sort of what we're interested in at the moment, but looking at other sensing modalities moving forward for sure. So with underwater images, you get huge variation in the characteristics, of course, due to depth, lighting, camera angles and distances, and then you introduce turbidity and blur into the mix as well. You have high levels of complexity. So you have a lot of overlapping, really poorly defined boundaries, and there's a real lack of that idea of objectness that I talked about before. So there is seagrass in this image, but you can't just point to it with one finger. It's kind of patchy and it's all over the ground and the carpet. Then we get into the ecology side of things. So you can have species that vary due to the season, due to the location, the environmental conditions. So it can be the same species, but it can look very different. And then you can have the opposite effect as well, where you have different species that look very, very similar. And basically the point I'm trying to drive home here is that you really do need to have, for the most part, someone who knows what they're doing, labeling these images, and being able to tell you which species are present. It's much harder for someone to come off the street and provide you with labeled data in this domain. Okay, so now to get into the work I've been doing. So starting off with this seagrass problem. So as you can appreciate, seagrass images are quite a complex example. So we have lots of leaves that are overlapping, growing together in a seagrass meadow. We're interested in knowing what different types of species are there because they have different abilities to sequester and store carbon. So it's nice to be able to delineate between these different species. It's just too costly and time consuming for an ecologist to label at the pixel level. I think even just what was done there was like 8 hours or something at work. So it's like pretty impossible. Instead, in my research, we came up with this idea of doing coarse segmentation. So having some grid over the image where we're classifying at that grid or patch level. And we argue that this is a good balance between still being able to tell what species are present and having some idea of relative abundance. And at the scale of a seagrass meadow, which could be in the tens of square kilometres or more, that's enough detail to be able to map that ecosystem. So we contributed the first data set in this space and sort of opened this up as a new task and a new direction of research. And we created these different classes which group together species of seagrass into what we call taxonomic morphotypes. With that work, we then did patch level classification. So this let us say in a bigger image how many patches belong to the different types of seagrass. And then in a more recent work, we wanted to take it a step further. So we wanted to say, if you actually just say what's in the image, so you don't actually provide the patch level labels, can you still get that coarse seagrass segmentation output and therefore reduce the labels you required by 96%. So in the work I'm just about to explain, what we're wanting to do is at training time have image level labels only, and at inference time still have that patch level inference. So we proposed a framework of an ensemble of 2 classifiers that were complementary and work nicely together. The first classifier we call CFeats, and it's based on contrasted training and feature similarity. So how it works is that we take every epoch a template for each of our classes. So we average the feature vectors across classes using just the image level labels only. And then once we have those templates for each of our classes, we can then take a query feature vector for our current patch and then find the cosine similarity between the current patch and the feature of that patch and the templates that we've already created, and use that to pseudo label the patch as whether it contains that type of seagrass or not. And we train that end to end. So it means that we're updating those templates as we go through the training process as well. Okay, and then our second classifier here was called CQUT. And for this one, we're leveraging the QUT large language model. So when QUT came out, it was kind of a big moment. And we were really interested to see whether it had good general understanding of concepts, which we could still leverage in some way in a very domain-specific task. So that's what we were trying to do in this work. And what we found is that it's really handy for those very general concepts. So QUT doesn't know the difference between Holophila spinulosa and Holophila ovalis. It knows nothing. But what it does know is, does this image contain seagrass? Does it contain sand, or does it contain water? And that's still useful for us because if we know at the image level what's in that whole image, we can then use QUT to tell us at the patch level whether it contains that seagrass or not. And so we can use that then to increase the amount of information that we have. And then we combined those in an ensemble and found that the 2 different approaches resulted in improved performance overall. We then deployed it on one of the surface vehicles. So here we can see it's classifying the sand as class 0 in pink and the type of seagrass as class number 3 there in yellow. Okay, so just to summarize, we've looked at this coarse seagrass segmentation task. We had the ensemble of classifiers, and what this allows us to then do is come up with ideas of relative abundance of different species and what the composition of a seagrass meadow looks like. In terms of the impact, there are lots of ecosystems, seagrass meadow ecosystems in Australia and the Pacific that haven't been mapped. And this is really important for those communities that rely on those ecosystems for their livelihoods. So if we know where the meadows are and we can map them, then potentially we can use that information to prevent dredging or damage caused by new infrastructure or these kind of things. Okay, so now on to the second body of work that I want to talk about today. So this is now switching for the coral segmentation problem. So here, the idea was that we have a whole lot of data already collected by ecologists over the last 20 years or so for their monitoring purposes. So what they generally do is they take images from a top-down viewpoint and they'll take static images along a transect. And these are called photo-quadrat surveys. They then take their top-down scaled nice image and they will label a certain number of sparse points. And it's typically arranged as a grid or it's arranged as just randomly distributed pixels. And they label just those points in the image and then they extrapolate that to work out what percentage of species are in that entire ecosystem. So this is something that the ecologists have been doing for years and years and years. And then sort of the computer vision community comes along and goes, okay, can we leverage this data in some way? What's a clever way that we can make use of it? Considering that it is quite a unique labeling regime, it's not something that we would really do on purpose in the computer vision community. So that's the sort of task that we're looking at. Obviously, with traditional semantic segmentation, we have training images, and for each training image, we have a dense ground truth mask that goes with it. So we normally have every pixel labeled at training time. For us, instead, we have the sparse points, and we want to introduce this point label propagation task. So this point label propagation task spreads out those sparse points in the image to create sort of a pseudo labeled mask that you can then use to train your semantic segmentation architecture. So previous approaches that have done that point label propagation have used superpixels, but the superpixels have only used RGB values. So they haven't considered the points themselves, and they also haven't used deep features either. So in this work, point label aware superpixels, what we did was take deep features within the image, and then we also introduced a new way of generating those superpixels such that similar pixels with similar features were brought together into the same superpixel region. And if we have a point label that has a different class label, they should be pushed into separate superpixels. And that means that you have superpixels that are informed by those points and should only contain one class at a time. So here we can see the superpixels at work, and the locations are optimized by that custom loss function. And from this, we were able to really improve the accuracy of that point label propagation task. Then in our more recent work, we thought, okay, that's great, but then you're relying on having those 100 to 300 point labels that the ecologists have already done for you previously. What if we actually wanted to do this task, but we only wanted to have 5 point labels instead? Or if you were going somewhere new and you wanted to very rapidly label some new species or a new location that you were visiting. So in this work, we were really trying to look at that extremely sparse label setting. So only 5 points per image, for example. In this work, we were excited by what we'd done with QUT previously and thought, is there another way that we can leverage these new advances in these big foundation models that are coming out? Are they going to be able to help us do this task? So we took the Dino V2 model. We actually used the denoise version, which works a little better. And we did a similar thing. So we created templates of a few labeled points that we have, and then we do some manipulation in the deep feature space so that we can cluster points to those templates that we already have. And it's a very simple idea, but it works really, really well. To go a step further, we also introduced this human in the loop framework. So with this framework, what we're doing is using the model to kind of propose to the human where would be the most beneficial location to put that point. So where in the image does it look a bit funny? Where in the image does it not match the previous examples that we have? There should be some uncertainty there, and so the human, would be well placed to put a new point in that location. So we use cosine similarity to our nearest known pixels. And that shows us the area in the image where it's most unknown. And then we can use that in combination with the distance map. So we don't want people to keep labeling in the same area in the image. We want to encourage a bit of exploration as well. And together we can then propose the next location for our expert to label. And then you can set some threshold of the number of points that you're wanting to label. And once you've reached that point, you have your augmented ground truth mask or your pseudo-labeled mask that you can then use to train your normal segmentation architecture. Just a little bit about that feature space that I'm talking about. So I think this is quite an interesting point. So in our work with the superpixels, we trained a feature extractor specifically for coral images. So we use some fully labeled data to do that task. And the features that we can see in the middle here are from that trained feature extractor on coral images. If we then look at Dino and whether it's the denoised version or not, Dino probably has had some training on some underwater images, but it hasn't been trained specifically on those. And yet it still performs better in the feature space at looking at which pixels should be within the same class. So here we can see that although Dino might not necessarily do a good job at segmenting a coral image itself out of the box, if you dig into the feature space and do some smart feature engineering with it, then it still is a very useful tool and something that we can use in a domain-specific task. So some of the results of this work, we did quite a comprehensive investigation into the number of labels that you need so that we can make some recommendations to people. People always ask me, how many labels do I need? What sort of labels should they be? What should I be doing to make it more efficient? So we did a bit of a study into that. What we can see is that if you have between 0 and 50 points in an image, the way that you do that labeling does make a really big difference. So if you use our human in the loop regime where we have Dino kind of helping us to say where to do that labeling, you get a significant improvement in intersection over union. So that's really valuable and really useful. If you've got 300 point labels in your image anyway, doesn't really matter. You can use this approach, you can use the superpixel approach that we previously proposed. You know, you've really got enough at that point to do a good point label propagation. And then just some examples there of the results that we're getting. So qualitatively, you can see even with 5-point labels, we're still producing a usable pseudo ground truth that we could train our model on. Okay, so just to wrap up, we did the superpixel work first and then considered that really sparse task and found that we could still do point label propagation even with only 5 labels per image. And in terms of the impact for this work, I think it's important to highlight that when we're doing coral monitoring in particular, there's more than 800 species of hard corals alone before you get into soft corals and all the other species that you'll encounter. So this is a really challenging task. I think what we need to be thinking about is how can we design methods that we can very quickly get up and running in a new location when we encounter some new species, and sort of maybe step back from this idea that we're going to have one model that works in the entire world for every type of coral that we might encounter. So that's really where this weekly supervised approach, these weekly supervised approaches come in and have their real place. And we were able to reduce those point labels significantly. Okay, so now moving on to what is the current and ongoing work that we're doing. So this is not fully polished and finished work by any means. I'm giving you a little bit of an insight into what we're doing right now. And this is more into the intervention space. So I think the monitoring piece is really important because we need to provide all of our experts with enough knowledge to make decisions on those interventions, but I'm also keen to see how can we actually assist in this reef restoration task. So to give you an idea of what we're looking at, so with the reef restoration and adaptation program, I talked a little bit about earlier how we can collect the coral spawn, put it in an aquaculture facility until it reaches a more mature stage of growth, and then they get inserted onto these ceramic reseeding devices, and these devices are designed to prevent fish from eating that coral spawn and to keep it nice and stable on the seafloor. And previously, they've been just manually deployed onto the reef in the order of maybe 500 last year. In the next few years, they're looking to really upscale this work significantly. They want to put out 20,000 of these devices in the next 2 years. So there's a big leap in terms of the order of magnitude of this task, and they're wanting to automate it with robotics, which is really, really good to see. So there's sort of a few different parts of this problem. The first part is this aquaculture stage. So instead of having our experts pull out the coral tiles and manually count the coral during that growth phase, instead we're using a robot arm equipped with a super high resolution camera to go ahead and monitor these tanks at certain intervals automatically. And then we can go ahead and use AI to automate the processing of those images as well. What's really challenging about these images is that the scientists would like to know things like when the coral is alive versus when it's half alive versus when it's dead. And those things are very, very hard even for a human to see. So that's something that I'll talk more about in the future work part of the talk. But yeah, there's lots of interesting problems with this task that we're still looking at. And then once we've got those baby corals and they've reached that stage of growth, then we're wanting to put them on those coral devices and deploy them back into the ocean. Obviously, that needs to be done in such a way that we don't waste the devices, and we also don't want to cause more damage in places where it's not suitable for that restoration to occur. So my part of the project is having a camera system mounted on a boat. That camera system is collecting images from the top down of the seafloor. We're wanting to operate into a depth of 10 meters. At the moment, this image, for example, is 4 meters. So we're dealing more with the shallow depths initially, and then we're working up into the deeper case there. And then we're wanting to decide whether or not to deploy based on that image. Based on my learnings with the seagrass monitoring, we've gone again for this patch approach because this is something we're wanting to do in real time as the boat is driving along. So we argue that the patches are probably a sufficient level of resolution for this task. And then looking at whether there's coral there, whether there's macroalgae which kills coral or not, whether the substrate is a suitable type for those coral devices to be dropped. And then as we move forward, using the patches as well is nice because it means that we can do deployment off either side of the boat based on whether one half of the image is more suitable for deployment than the other, for example. So our first field trials of this work we did in September in the Whitsundays. So here we're just doing the frame, we're doing frame level deployment based on the percentage of patches that are deemed suitable or not suitable. And that's a tunable threshold that we're still working out based on how certain the ecologists want to be about that deployment. And then we can track where we're deploying those corals using GPS so that the next year they can go back and monitor those same devices and check how they're faring. Okay, so that's the sort of 3 main bodies of work that I wanted to touch on today. Just a little bit now about what I think are still quite unsolved problems and things that I'm keen to look at more in the future. So the first one is, of course, turbid water imaging. So everything that I've talked about so far, obviously we are using more of those RGB cameras. So if you do have a really poor visibility day and there's a lot of turbidity in the water, it's a big problem. And so it limits the operating conditions of the monitoring or the reef restoration approach that you're trying to do. As I mentioned earlier, this is primarily because more people have RGB cameras. So we don't want to necessarily dive full bore into these other sensing modes, but I do think there's a really important place for them, and it's something that I'm keen to investigate more. And then the next one is this fine-grained recognition task. So I talked about it briefly with the coral babies, but it happens a lot in ecological tasks that you have some variable or some factor that you're interested in measuring that you can't neatly put into a category. And a lot of our computer vision approaches are really good for classifying things into classes. But I think that we can do more in terms of continuous ecological indicators. So that could be how densely the seagrass is growing, or how alive or dead something is. And you can think as well, for example, coral bleaching. When a coral has been bleached, it's not necessarily dead. It might die, but there's a spectrum there as well. So I think that that's an interesting task I'd like to look at. And then the next one is looking more at these large language models. So again, they're great for general concepts and we can use them in a whole range of tasks, but it would be really nice if they had more of an understanding of specific ecological terms and descriptors. So that maybe an ecologist could say to the large language model exactly what they're looking for, and then have that be converted into some embedding space that enables the model to then to do open set recognition of different species. And you know, one of the examples of when this would be useful is in these aquaculture tanks. You can have pests that show up that destroy all the corals, and that is devastating and very expensive. But you don't know what the pest is going to be before it rocks up. So you don't have training data for it in advance. So, you know, either it's another small data weekly supervised problem, or maybe if we can use like prompting based on domain specific terms, maybe that's another way that we can do that. Okay, that brings me to the end of my talk, but I would love to answer any questions or to have any conversations about what I've presented, and I'd love to hear your ideas as well. Thank you so much.
Any questions? Yep.
Yeah, fantastic talk. Thank you very much. I have a question kind of about, let's say, the bigger context of this in terms of, let's say, you can use robotics to collect lots more information and use all sorts of segmentation to generate lots more data from that, lots more information. In the context of this being used downstream by policymakers or some other decision makers to make better decisions, do you think they're equipped to deal with the kind of information you're providing, or is it providing the same information they would normally look at but more accurately or more cost effectively, or is it just going to be like they look and say, what's all this? I don't really know how to deal with this.
That's a great question. So what I would say, so for example, the seagrass monitoring is probably the best example of that. On the Sunshine Coast, they need to monitor their seagrass meadows very regularly, even weekly or monthly, because they need to know where they can dredge, because they're constantly dredging along the coast. So for that case, it's like, can we send out a platform that quickly monitors the seagrass and says, quantitatively, is it more or less than it was last week or last month when we were there? And then obviously give them some interpretable, understandable assessment of that. So I think you're right that there's a step between what I do and what they need. There needs to be something that is done in the middle that gives them information that they can rapidly act on. But I think that step is, it's definitely a practical, meaningful step. And this is something that they actively need constantly. So I think that we can definitely help them tackle this problem, but you're right that there is a little step in between where we need to interpret the results and give them some sort of idea of whether the seagrass is better or worse where it is. Maybe a map, for example, the heat map that visually depicts that would be a good way to do it. Yeah.
I guess the flip side is they have to make those decisions now with very little information.
Yeah.
So there's any extra information is probably useful. I was going to ask you, you had with the sort of human in the loop thing, you had a notion of the uncertainty of the labeling that you're then using to choose what next. Do you use any notion of that uncertainty in training the models? So do you push that uncertainty through
We're not
the classifier?
currently doing that, but that's a very good idea. And in fact, an idea I've grappled with before as well is if you've got some points that the ecologist has told you what the class it is. And then you've got other points in the image where you've pseudo-labeled them using your approach. And I think what we could do there is increase the weighting of the points that have actually been given to us and decrease the weighting of the pseudo-labels. So yeah, I think there's definitely a lot more in that area that we can delve into. Yeah, I agree.
Thank you. Thanks, Scarlett. Really rather interesting. I was thinking about your pushing the RGB aspects of all of your monitoring. it seems to be sort of narrowing your sensing modalities a bit, because I mean hyperspectral and multispectral cameras are really easily available these days. So would that make it maybe your classifications much easier if you look at the other spectrum?
I personally haven't done a lot of work in that space, so I don't want to sound like an expert on it, but I understand that, especially for corals, that the signatures in hyperspectral and multispectral imagery does make it easier to tell the difference between those species. Again, correct me if anyone knows more, but I do understand that. And I think it's something that we will continue to look into. I think it's more from the standpoint that, a lot of the time we have citizen science groups that have GoPros and they're just out there snorkeling, collecting imagery that way. So we want, in this work particularly, we were wanting to produce algorithms that could be readily used by anyone. But I completely agree with you that these other sensing modes can give us a lot more in terms of information. Yeah, I think it's a balance.
I think you also have to be a little bit careful about modeling the inherent optical properties of the water because they're very wavelength dependent. So if you're making classification at individual wavelengths, it's very dependent on what the water is doing
at that time. Yeah.
So you have to be a little bit careful.
I was really, with your future work, is around like continuous classification of like problem. I feel like there'd be massive like cross pollination between like people do like agricultural work trying to figure out is this fruit like right or not.
Absolutely.
And I know, remember Chris Leonard was trying to do a bit of work in that space. How much cross pollination is there actually in terms of like where you're thinking of going like classifying coral versus like classifying fruit? So do you think like the methods be the same?
Yeah, there's a whole lot of overlap there, which I think is really cool. That particular idea came out of the discussion with researchers in our group that are doing monitoring in Antarctica. So they're using drones and they're looking for like mosses and lichens that grow together. So it's the idea that you can't necessarily say that's moss, that's lichen. It's kind of like certain percentages of moss and lichen, and then they're different levels of health as well. So they also want to know that half dead, alive, so there's all of these different continuous kind of spectrums that we're trying to work through. So I think that the ideas that we have in our particular use case are definitely transferable to other problems as well, for sure. In terms of the amount of collaboration that's going on, in our lab, we're always talking to each other between the teams, but we haven't set up like a formal project for that work yet. But yeah, you're right, it could be definitely different fields.
I'd just love to know about like, how do you intend like approaching the problem? Like, do you think it's just like a case like more labels and like a better set of classification? I think maybe like a new novel approach.
It's you tell any ideas? Tell me. Tell me more. I think they'll definitely will need to have some way of identifying the extremes of that spectrum, I suppose. So I think we'll definitely have to have some way of grounding it as like, this is what it looks like when there's fully dead, and this is what it looks like when it's fully alive, and somehow going from there. But exactly how we train this, exactly what it looks like, it's open research problem. So happy to discuss how we go about that.
Problem with geology as well, where you take especially with hyperspectral imagery of like a rock, and the rock is made up of many different minerals, and each one has a certain sort of signature, and you want to figure out what the percentages are. That's something that's studied quite a lot.
Okay, I'll be looking up the geologic literature after this.
But it's more of I think it's more of a regression problem, rather than classification.
Sure.
You've got continuous outputs.
Yeah.
It's also blind source separation in that case. You have all those kind of different mixed signals, and they're trying to.
Thank you for the talk. So the way to obtain superpixels from points, points generated by experts, and then you obtain semantic labels from, right? So this reminds me of gospel points from segmentation. Models such as segmentation, for example. Would it be a good person? Where
Yeah, really nice question. So the point aware superpixel work predates segment anything. So let me just point that out first. And then Segment Anything came out. And also Segment Anything 2 has also come out. And we've tried that quite a bit on our images. What we find is a lot of the time, the poorly defined boundaries of what we're looking at ends up breaking Segment Anything. So in some cases, it works nicely, especially if you have, for example, an image and there's like a brain coral over here and like a plate coral over there, it can sort of find the boundary of that. If you're sort of more zoomed in and everything's overlapping and the boundaries are not so clear, then it does really a very poor job of creating those segments. So I think there's still a place for some custom approaches in this problem, but I think we can make use of Segment Anything to help us for sure. It might just not be an out of the box immediate plug and play.
Can it be because of the main change, for example, for strains?
Yes. Yeah. Yes. Yeah. And also if your image quality is not amazing, if you've got turbidity or other effects, then Segment Anything kind of gives up as well. It'll just produce big segment for the cloudiest part of the image, for example, and it doesn't look through that at what's underneath. So yeah, so I think it did change the game a bit when Segment Anything came out, but I would say it wasn't like I was completely out of job the second that happened. Like there's still more that we can do. Yeah. Yes.
Just combining the last two sort of questions and the continuous variable stuff with the segmentation, does it mean that what we actually want to do is not really segmentation anyway? You want to more like establish densities of different types in an image?
Yes, and that's the end game, right? Is to have these relative abundances of the different species, so that the scientists can use that to establish the overall coral reef health. Exactly how we arrive at those estimates is still completely up for debate. Yeah.
Question.
Yes.
Yes. So you mentioned you use Reef model.
Yes.
Some adaptation and technology. Let's say you're using not local or not private today. This I would ask for different types of applications, like not that of the
Yes.
but more temperature of the sugar that like
Yeah. So we did those experiments in that paper. So we would, when we tried a whole lot of different prompts as well. So we would say a plant with round leaves, a plant with long skinny leaves, an underwater plant with round, a seagrass with round, we just did all of the different possible like natural language descriptions of the classes we were looking for. And it couldn't discriminate between that level of detail. It might be since, of course, that came out a few years ago now. So maybe there are other vision language models out there that are better at dealing with these sort of general descriptors. So maybe if we redid those experiments today, we would get a slightly different result. But at the time, it wasn't able to do it. And the other reason I would point out is that a model like QUT is quite resource heavy and slow. So if you wanted to use QUT directly in real time on a surface vehicle, you would struggle. But if you use QUT to label or to train a really lightweight classifier, then you can just deploy that straight away and it's easy. So yeah, I think we should think about the other considerations of the task as well. Okay. Oh, yes, sorry.
So about the technical thing, not the analysis of the data, but about the extraction of the data. So you presented an alternative technique with some probably when it's not the image is not clear. So in this case, might be somewhat to identify some type of conditions. So probably in this case, we will not have a representation of what is happening by image. So mostly the parts like fine patches that's the propagation of weight. So this is another thing about
Yeah, I would say I'm only dipping my toe into the other sensing modality, so it's something that I don't know a huge deal about yet. So yeah, it's really
So
just
there's a community on that you guys contact.
okay. Oh, fantastic! I will definitely look at that. Yeah, we have a few ideas of different combining different sensors together and seeing where we can go with that. But yeah, no work has started yet in that direction. Yeah.
But do you have any questions online? No. Right. Thank you.
Thanks.